Hi there. Welcome to Labster. In this lab, you will learn about the general structure of a bacterial cell, and how this structure can help bacteria survive in extreme environments, such as Antarctica. This simulation uses voiceovers. If you don't have audio, click the speaker symbol in the top right corner of your lab pad to switch the play mode. If you disable the sound effects, the lab pad will pop up whenever there are instructions to read. Select the audio setup you prefer. To mute individual audio options, simply select them. Click Continue to learn how to navigate through your mission. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a Labster simulation? Perfect, then let's get started with your first mission. Our friend Nicholas is a researcher in Antarctica. He has collected a sample of ice that contains several types of bacteria. These bacteria have survived thousands of years in extreme conditions. Isn't it strange that bacteria can survive for thousands of years while eukaryotic cells cannot? Nicholas wants to know why the bacteria can survive. Let's have a look at the Vibrio bacteria and compare it to a eukaryotic cell, in this case from a human. Go to the hollow table to inspect the Vibrio bacteria. Before we start, we should make sure we are on the same page. Which of the following is neither a eukaryotic nor a prokaryotic cell? That's right. This answer might surprise you. Viruses are not cells at all. They lack the characteristics of living organisms, except the ability to replicate. However, in order to replicate, the virus needs to infect a living cell. Click on the bacterial cell surface. Check out the screen showing the bacterial cell wall. Raise your lab pad to continue. Bacteria usually have either a gram-positive or gram-negative exterior surface. This cell's wall has a thin layer of peptidoglycan and has an outer membrane, meaning it is a gram-negative bacterium. You can read more about the different cell walls of bacteria in the theory pages. You can view the image displayed on the screen using the View Image button. What is the main function of the cell wall? That's right. Did you know that some bacteria don't have a cell wall? A common one is Mycoplasma bacteria. Mycoplasma is a special risk to lab work. They are hard to detect and hard to get rid of because they are so small and don't have a cell wall, which is often a target for antibiotic treatments. Click on the eukaryotic cell surface. Check out the plasma membrane on the screen. Raise your lab pad to continue. This eukaryote's external structure is a plasma membrane. Some eukaryotes though, like fungi or plants, do have cell walls. This outer barrier consists of a lipid bilayer with multiple embedded proteins that serve different functions in molecular transport and signaling. Overall, the structure and function of prokaryote and eukaryote plasma membranes are very similar. You can view the image displayed on the screen using View Image button. Both eukaryotes and bacteria contain plasma membranes. Which of the following statements explains the function of the plasma membrane? Excellent. You are too smart. I'll be out of a job soon if you continue. Yes, the plasma membrane is important for the transport of ions, nutrients, and waste across the membrane. Now we know the difference between the outer structures of an eukaryotic cell and the outer structures of the bacteria. Let's focus on what we see inside the plasma membrane of the bacteria and the eukaryotic cell. This area is called the cytoplasm and contains various organelles and cellular structures, depending on cell type. Organelles and other cellular components are structures within a cell that perform specific functions like storing nutrients, producing energy or containing the genetic code. The hollow table has a mix of eukaryotic organelles and bacterial cellular structures. Can you place them into the right cell? Organelles and other cellular components are structures within a cell that perform specific functions like storing nutrients, producing energy or containing the genetic code. Pick up the different organelles and cellular structures on hollow table and place each one into the eukaryotic or the prokaryotic cell dependent on which cell it belongs to. If you are missing space inside the cell, check if you have placed an organelle or cellular structure wrongly into that cell. Grab it inside the cell, place it back on the hollow table and add it inside one of the two cells again. There might be excess organelles and cellular structures on the table. You can always check out the theory button on the top left part of the lab pad if needed. Place the organelle or cellular structure inside the correct cell. Absolutely. This is an organelle of the eukaryotic cell. A pretty advanced organelle actually. Let's not go into details, but it's basically a network of membranous tubules which is continuous with the nuclear membrane. It usually has ribosomes attached and is involved in protein and lipid synthesis. That's right. The nucleus is where we find the eukaryotic DNA. Busy nailing those components of the eukaryotic cell. The Golgi apparatus is a complex of vesicles and folded membranes. It's involved in secretion and intracellular transport in the eukaryotic cell. You nailed it. The nucleoid contains the DNA of bacteria. It's double-stranded and usually circular, but this can vary, as can its length, between bacterial species. The genetic code itself can also differ between species. Awesome, nailed this one. Plasmids are extra chromosomal DNA that can be found in bacteria. They are small, circular, and double-stranded pieces of DNA that usually provide a genetic advantage for the bacteria. For example, they can carry resistance genes or other genes that make the bacteria adapt to the surrounding environment. Plasmids are also a handy tool when cloning, transferring, and modifying genes in the laboratory. Yes, the mitochondrion is a part of the eukaryotic cell. Mitochondria are the power organelles of eukaryotic cells and are not present in bacteria. Actually, mitochondria are believed to originate from bacteria and were incorporated inside eukaryotic cells. For example, each mitochondrion has its own circular DNA, similar to what we see in bacteria. Pretty cool. Correct. There can be numerous inclusion bodies, or granules, in the bacterial cytoplasm. These bodies serve as storage vessels for many different molecules, including nutrients and factors for extracellular release. Fantastic. Yes, the bacterial ribosome consists of two subunits, a small 30S subunit, and a larger 50S subunit. Combined they form the 70S ribosome. Each bacterium contains several ribosomes in their cytoplasm where they translate the genetic code of the bacterium into proteins. The ribosomes are similar across prokaryotes. You definitely know your cell components. That is correct. The lysosome is part of the eukaryotic cell. It's a membrane-bound organelle which contains enzymes that can break down many kinds of molecules. That's right. The 80S ribosome is present in the eukaryotic cell and not in the bacteria. We found a few different bacteria in our sample from Antarctica. Let us compare the Vibrio bacteria to the common E. coli that was in our sample, and another species of bacteria that live in a different environment. Their similarities and differences might give us some idea as to what explains the survival of the Antarctica bacteria. This is E. coli. It's one of the most studied bacteria, so you might have heard of it. It can cause diarrhea if we get infected, but it also lives naturally in our intestines where it helps process the nutrients that we eat every day. Have a look around in the E. coli to see how it differs from the Antarctica bacteria. Find out more about the E. coli bacteria by clicking on its various components. This is the capsule of E. coli which surrounds the cell wall. It consists of long chains of polysaccharides. The capsule protects against host defenses and environmental stresses. The hair-like appendages, fimbriae, are important for E. coli to stick to surfaces like the intestinal lining. The flagella is used by E. coli to move rapidly towards attractants and away from repellents. The cell wall consists of a thin peptidoglycan layer and an outer membrane, meaning that E. coli has a gram-negative cell wall. This F plasmid, also called fertilization plasmid, contains genes that allow the plasmid DNA to be transferred between multiple E. coli bacterium. This R plasmid contains resistance to ciprofloxacin, which is a broad-spectrum antibiotic used to treat E. coli infections. This is the DNA of E. coli. It is approximately 4.7 million base pairs long, circular, and we think there are more than 4,200 encoding sequences. There are probably more, and this varies slightly between different strains of E. coli. These are the 70 S ribosomes essential for all prokaryotes for translating the genetic code into proteins. In bacteria, mRNA is translated into protein as soon as it is transcribed. Click on the right arrow on the hollow table to explore a bacteria from another extreme environment. This type of bacteria is a cocci. It's been found in a hot spring environment. Have a closer look at the inside and outside of the bacterium to compare it to the bacteria we found in Antarctica. Find out more about this coccus bacterium by clicking on its various components. This is the capsule that protects the coccus against the external environment. It also contains fimbriae so the bacteria can stick to surfaces in the hot springs. If you have ever been in a river and stepped onto rocks, you would have felt that rocks can be very slippery with a moist-feeling layer covering them. This was most likely a lot of cocci sticking onto the rocks. As you can see on the screen, this cell wall doesn't have an outer membrane but has a thick layer of peptidoglycan. So this cell wall is different from the Vibrio bacteria we found. This bacteria has a gram-positive cell wall. The thick peptidoglycan layer makes the bacteria resist the high extracellular pressure, ensuring that the cell doesn't burst. To survive hot springs, the coccus has gas vacuoles that keep it orientated and buoyant in the water. These are 70 S ribosomes essential for all prokaryotes. The DNA of the coccus is much longer than that of E. coli. We haven't studied all the proteins encoded by the genes, but a fair amount of them code for proteins that are essential for living in the hot temperatures. These inclusion bodies contain nutrients that the coccus needs to survive when nutrients are scarce. We need to be a bit careful about this plasmid. It encodes for a toxin that can be harmful to animals and us. This type of plasmid is called a virulence plasmid. Click on the right arrow on the hollow table to have a look at the Vibrio bacteria we found in Antarctica. Let's have a closer look at the Vibrio bacteria that is in our sample from Antarctica. We have been through quite a lot of different components for the different bacteria. Let's apply this knowledge here and see how the outer and inner structures differ, and how they may play a role in helping the Vibrio bacteria survive the extreme conditions of Antarctica. Find out more about the Vibrio bacteria by clicking on its various components. This cell wall is gram-negative, as we've already seen. The cell wall protects the bacteria. How does it protect the Vibrio in Antarctica? That's right. The cell wall protects the loss of water from the cell or the gain of water into the cell. Too little or too much water can harm the bacteria. How does the ribosome ensure that the Vibrio can survive in the extreme environment? Absolutely. The ribosomes are essential for bacterial survival. Without the ribosomes, the genetic code would not be translated. This means no proteins, which ultimately means no cell. If we have no proteins, we have no cell survival. The DNA is circular, similar to the other two bacteria, but is 3.3 megabase pairs long. I am not sure how many proteins are encoded by this bacteria since we have only just discovered it. However, some initial studies showed that it contains some genes that encode for so-called antifreeze proteins. These proteins ensure that the bacteria is still viable even at freezing temperatures where the bacteria would normally be completely frozen. How is the nucleoid involved in helping the Vibrio bacteria survive the harsh environment? Yes, we don't know much about the Vibrio DNA yet, but from initial studies, we have found that it encodes for antifreeze proteins which prevent the bacteria from freezing. That's pretty awesome. The inclusion bodies hold carbon resources that can help the bacteria survive under nutrient-scarce conditions. How do the inclusion bodies assist the bacteria in survival? Nailed this one. Yes, the inclusion bodies hold nutrients that the bacteria can use when nutrients in the environment are scarce. This helps the bacteria survive extreme conditions. This plasmid contains genes that code for bacteriocins, which are proteins that can kill other bacteria and thus defend the vibrio. What is the plasmid's role in survival in extreme conditions? That's right. Having this plasmid gives the vibrio bacteria an advantage over other bacteria. Do you still want to examine the bacteria? Are you done examining the bacteria? Great. Then let's carry on with this experiment. Well done. You have completed the simulation. You have really mastered an understanding of the bacteria's outer cell structures and inner cell structures. Also, we learned how this structure helps bacteria to survive in extreme conditions. The information you collected has been relayed to Nicholas. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.